I've been hearing for years about the benefits of a plant-based diet, but really, we can get remyelination help from flowers? Well, hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam, and I've been living with multiple sclerosis for almost 40 years. I know that you may be tired of hearing me extol the virtues of Oregon Health and Science University, but I'm gonna do it yet again because new research coming out of OHSU in conjunction with the University of Portland, Oregon, has discovered that there are compounds in flowers that actually might assist with remyelination. And of course, we want to know more about that. So let's look at the article. This article first appeared just a few days ago on the OHSU News website. It's called Natural Compound Found in Flowers Halts Progression of Multiple Sclerosis and Some Cancers. OHSU-led discovery benefited from years of study by the University of Portland undergraduates. And this is an interesting idea, too, that when one research team discovers something, it's really nice when another one who has perhaps more access to research dollars picks it up and takes it and runs with it. And I think this one could actually amount to something. As the photo describes here, new research led by OHSU finds that a type of plant-derived chemical compound found in flowers may be useful in treating cancer and demyelinating conditions like multiple sclerosis. Oh, we have to know more about this. The article says that scientists have identified a natural compound that halts the process involved in the progression of certain forms of cancer and demyelinating conditions, those that damage the sheath, known as myelin, that surrounds neurons, such as multiple sclerosis. A study published online today in the Journal of Biological Chemistry found a plant-derived flavonoid, a type of chemical compound called sulforaphane, blocked the activity of an enzyme involved in MS and cancer. The discovery was made in cells tested in an Oregon Health and Science University laboratory. The next step is to test the compound in animal models to ascertain its effectiveness and possible side effects in treating cancer and neurodegenerative conditions like MS. We think this is a drug that could have impact in a lot of different areas, said Larry Sherman, PhD, professor in the Division of Neuroscience at OHSU's Oregon National Primate Research Center. The researchers found that sulforaphane, along with a pair of synthetic compounds that were also tested in live cells, inhibited the activity of a particular type of enzyme known as hyaluronidase, which naturally degrades hyaluronic acid. That's important because when hyaluronic acid is broken down into fragments, it is known to cause problems in at least two ways. First, it forestalls myelin repair. It prevents the maturation of oligodendrocytes, which are cells that produce myelin. Myelin is the protective sheath covering each nerve cell's axon, the thread-like portion of a cell that transmits electrical signals between cells. Damage to myelin is associated with MS, stroke, brain injuries, and certain forms of dementia. In addition, delay in myelination can affect infants born prematurely, leading to brain damage or cerebral palsy. Number two, it allows cancer cells to proliferate. In cancerous tumors, hyaluronidase activity can allow cancer cells to proliferate unchecked by normal cellular death. Now we have an inhibitor that could actually stop that, says Sherman, who is also a professor of cell developmental and cancer biology in the OHSU School of Medicine. The new research focuses on inhibiting a specific type of hyaluronidase known as cell migration-inducing and hyaluronin-binding protein, or CEMIP. Now keep that acronym in mind because you're going to see it again throughout the article, and it's kind of a tongue twister what it stands for, so just keep it in mind and refer to it later. In addition to MS and cancer, CEMIP is implicated in a range of disorders, including osteoarthritis, which I have, skin infections, brain injury caused by heavy alcohol use, and possibly other neurodevelopment disorders, including Alzheimer's disease. The study indicates its activity appears to be inhibited by sulforaphane molecules in flowers. The discovery came after years of undergraduates painstakingly screening plant compounds in the lab of co-author Angela Hoffman, PhD, a longtime and now retired professor of chemistry at the University of Portland. Over the years, her students have been grinding up these flowers, extracting molecules and testing to see if any of them blocked hyaluronidase activity, Sherman said. Finally, a couple of years ago, they found a compound that was promising. Alec Peters, a graduate student in Sherman's lab at OHSU, found that this compound blocked CEMIP activity in a tumor cell line and in oligodendrocyte progenitor cells. Oligodendrocytes generate myelin, and we've certainly been seeing that in a lot of the videos that I've been posting. Hoffman, a nun and chemistry professor, who earlier this year retired from the University of Portland after 35 years to lead her convent, began collaborating with Sherman a decade ago. Over that time, she said, hundreds of undergraduate students worked on breaking down dozens of plants to their molecular essence and then testing to see whether any of the compounds worked to neutralize CEMIP. The new publication validates the students' diligent work over many years, she said. Directing the students to be able to do this kind of research helps them in their careers, Hoffman said. This discovery could be useful for Alzheimer's or other neurodegenerative conditions, as long as the underlying problem relates to hyaluronic acid being broken apart. This could be helpful for people. And I'm going to read the funding paragraph just so that you can see where the funding for this is coming from. The research was supported by the National Institutes of Health Grant Award, and there's the number, for the operation of the Oregon National Primate Research Center at OHSU. Congressionally Directed Medical Research Program Grant Award, and there's the number. The National Multiple Sclerosis Society, and there's the grant award number. The National Institute of Neurological Disorders and Stroke of the NIH, and there's the grant award number. And the National Institute of Alcohol Abuse and Alcoholism of the NIH, and there's the grant award number. The content is solely the responsibility of the authors and does not necessarily represent the official views of the NIH. That is interesting just to see that there are so many different entities, though I admit most of them are government. But the National MS Society is involved in this as well. So a number of entities are believing that there's some promise here. Remyelination, as you know, is a very hot topic right now. And this one must seem at least a little bit plausible, or they would not be funding it. So I am looking forward to finding out more about this. Oh, wait, hold on a minute. By doing a little more looking around, I found news reporting on this from KOIN in Portland. So I'll play that, and at least you'll get to see the researchers that are leading this effort.
Welcome back. You've seen these, right? Dahlias are everywhere here in the Pacific Northwest, brightening even the rainiest of days. Well, now researchers at OHSU found a game-changing compound in these vibrant flowers that could fight cancer and other diseases. This breakthrough shows that hope can bloom in the most unexpected places.
We can grow live cells, live nerve cells.
In the lab at OHSU, Dr. Larry Sherman and his team are unlocking secrets from dahlias.
These are all different types of cells.
The results could change cancer and neurodegenerative disease treatments forever.
It's putting that puzzle together that's really exciting.
Years of dedication led to this breakthrough, thanks in part to a nun turned professor.
That's the coolest part of doing I don't know what's going to happen next research. And that's the kind I like.
Dr. Angela Hoffman and her students from University of Portland turned pretty flowers into a powerful discovery. Their years of work on orange and yellow dahlias led to sulforaphane. More on that in a moment. Now she sees them in a whole new light.
I say thank you for your pigments. I love it.
So here's how it works. When inflammation triggers c-Myc production, it breaks down hyaluronic acid, making nerve repair harder in MS and helping cancer cells grow. But sulfuritin, that key ingredient, blocks c-Myc, boosting nerve repair and making it tougher for cancer cells to thrive, offering new hope for treatment. And it's not just MS and cancer. Researchers say it may also treat Alzheimer's, other brain injuries, and even cerebral palsy.
It's called a wet lab space. So we do a lot of molecular biology and biochemistry here.
From the lab to the next phase, Dr. Sherman says the goal is to test sulfuritin in animals to see how it works. Just one step of many toward potential new treatment options.
If we can do anything that's going to help that group of patients in any way, it would blow my mind. It would be awesome.
It's important to note here, both doctors stress that while dahlias are beautiful, they're not for eating. The key here is a specific compound from that flower. From here, though, medical chemists will work to turn it into a potential drug.
If I find out more information, you know that I will tell you about it. And if I find out that there's a clinical trial going on, I'll certainly let you know about that too. But it sounds like this could be an interesting breakthrough because once again, the answer to these very complicated questions might be as simple as something that you can grow in your own garden. Well, that's all I have for you today. So until my next video, please do take really good care of yourself. You don't want to be too laid up to take advantage of these new developments that are coming down the pike. I'll see you again soon.